All right. So this will be fine. We have plenty of time until 11. I don't
Yeah.
think we'll spend that much. So I wanted to just go over the count normalization details with you guys, because especially because there's some confusion as to when do I use normalized counts and why should I give raw counts to the differential expression analysis methods. So we'll go over it in detail, and if you guys have any questions, please ask. All right, so let me start. All right, so because I noticed that it generated some confusion when I was talking about quantification, I wanted to review a little bit the concept of relative and absolute transcript levels. So when we talk about relative abundance of a specific transcript, what do we mean? So we mean the percent composition of that particular transcript with respect to the total number of transcripts that are present in your sample to begin with. And let's put it into an example. So we have, for example, 10 million sequence reads in a given sample. And out of those reads, 100,000 of them mapped to gene A. So that would mean that 100,000 over 10 million, 1% of the reads come from gene A, and that is the relative abundance of gene A. And you may think that I'm exaggerating with this example of a gene that consists of 1% of the reads in your sample, but actually that happens. So this is an example of the expression in beta cells from pancreas. And here I'm showing different genes, and in red you see the expression of insulin, for example, in the beta cells compared to the alpha cells. And as you can see, around 12% of your mapped reads in beta cells come from insulin 2, from the insulin 2 gene. So this is actually a huge problem when you're doing analysis in beta cells, because that means that the depth of your sequencing needs to be large enough so that the majority of the reads you capture doesn't just come from insulin 2, but in fact, there's other transcripts sampled in there as well. So just to show you how it actually happens, and this is would be the relative abundance of insulin 2, it would be around 12%. All right, so one thing you guys also asked me was, can we ever get absolute quantification? Okay, I know that 12% of my sample is insulin 2, but can I actually know how many molecules of insulin 2 are present inside a beta cell? And the answer is yes, you can, but it will require a lot more information, and it can't be done with just the simple RNA-seq experiments that we've been talking about. So you can have one of two things. The first one is, if you actually know from the literature what are the values for the mRNA content of a given cell. So for example, there have been studies with hepatic cells where you actually know on average if a liver cell, how much a liver cell expresses in terms of total RNA. So you know how many molecules of mRNAs will be present in that cell. Or another option is you know exactly the starting number of cells you used in your experiment. So let's say it came from FACS sorting, just like this example that I'm showing you for beta and alpha cells. And during FACS sorting, you know exactly how many cells were positive for your marker, and you separated them into a tube, and that's where you performed your RNA extraction. So you still need to know what are your RNA extraction yields. So you will lose some RNA molecules during the extraction process. But if you know that and you combine it with the fact that you know exactly how many cells you started with, then yes, you can try to get back at the absolute quantification levels. But in both of these cases, you will require something else. And that something else are spike-ins. So these are molecules or RNA standards that we know their length, and they have variable lengths. Ideally, you have a combination of spike-ins that have variable lengths, that have variable and known GC content. So you want to simulate how you would lose particular transcript molecules during the library preparation process, and you know the input amount of these RNA standards that you spiked in your sample. So you perform this spike in right at the beginning of your library construction, and then by the end of it and after sequencing, you can try to deconvolute the losses that you experienced from the technical preparation and get back to the absolute levels using one of these two known quantities. So either you know how much transcript, how many mRNA molecules you expected in that cell to begin with, or you know the starting number of cells and the RNA preparation yields. So I hope that's clear and resolved some of the doubts you guys had before. And let's do a quick.
Can I ask you a question
Yeah.
about that slide before you go on?
Yep.
The absolute quantification was clear to me. I understood that, but I'm still thinking about the example of the insulin gene that you gave us with 10 million reads and 100,000 reads mapped to gene A. So you have 1% from gene A.
Yeah.
Is there not a correction for the length of gene A?
Yes, there is. We'll go through that.
Okay.
But I'm just so in this case, I am not comparing gene A to gene B. So that's where the length will come into. So that's exactly a good segue into what I was going to say about normalization. So it depends on your goal, right? So if you just want to compare gene A across different cell types, for example, here's what I'm doing with beta and alpha cells, then I don't really care about the length because it's the same in both cells. It's the same gene.
It's just
right?
insulin too in the red cells and the green cells.
The relative abundance of the gene just in respect to how many transcripts you have in the cell to begin with. Okay.
Okay.
So let's.
Yeah, that makes sense.
Yeah.
Thank you.
So let's continue with that. So basically, your RNA-seq read output, just to recap what we talked about before, is going to depend on the transcript expression level in the cellular tissue. So is it a highly expressed transcript? That means you're going to get more reads in your RNA-seq experiment. Is it a longer transcript? That means you also will get more reads. And what is the depth of sequencing you have to begin with? So these are the three different variables that we want to control for when we talk about normalization. So what are these methods of normalizing? So one of the most common ones, and it was used for a long time, has been FPKM or RPKM. So what does it mean? It means fragments or reads, so that's what the F and R stand for, per kilobase, so every 1,000 bases, per million mapped reads. So let's go back a little bit and remember how we sequence these libraries to begin with. So what does it mean to be read or fragment? So it depends on the fact that you either sequence both ends of your fragment in your library or only one end. So in the beginning, most of the libraries used to be single end. And that means that you ligated adapters to this fragment, but you only sequenced one end of this fragment. So you could have reads per kilobase per million because each read came from one fragment in your original library. But in the case of paired end libraries, if we used read information, we would be artificially doubling our read counts. And that's because we sequence both ends of the fragment. So what matters is actually the pair of reads and not each read by itself. So they renamed this method to FPKM so that you know you're not talking about individual reads, but you're talking about how many fragments from your original library you have a representation for in your sequence reads. So I hope that makes sense. And that's the only difference between FPKM and RPKM. So most of the time I'm going to talk about fragments because if it's single end or paired end, it doesn't matter. What it means is if you see that read from the pair or not, you had one fragment in your library. So this method can account for these two things. So both the transcript length and the library sequencing depth. So it will take care of both, because you're dividing the number of fragments you see by the length of the gene and by the total number of mapped reads you have. And here's just to illustrate how you would get it from the table you guys have been working with. So you have the counts and you have the FPKM for the FGF21 gene. And like Alper was saying, in this case, normalization didn't really show a dramatic difference because these samples were sequenced at the same depth. So you see 173 reads in the control and 332 in the experimental condition. And when you normalize that to FPKM, the numbers change, but it's actually kind of the same. It's a little more than twofold difference from the control to the experimental condition. But in cases of imbalance in the sequencing that you did, then it will matter a lot. And last thing I want to show, oh, no, that was too far. Okay, and the last thing I want to show is just the complete formula. So I've already explained it to you guys by saying it out loud, like what does this mean? This is just to put the actual numbers there. So what you do is you divide the number of reads you obtain for a gene by the length of that gene. And because genes have different sizes, that means for one gene, you're going to be dividing the number of reads by 700. And for another gene, you're going to be dividing the number of reads by 1,500 bases. So that would give you reads per base. But we want to make that number a little larger and say for every 1,000 bases, how much do I expect? So that's why we multiply by 1,000. And all of this would be divided by the total counts, which would tell you how many reads to expect from the total number of the relative abundance compared to the total number of sequence reads. And you multiply by 1,000 because otherwise this would be a very small fraction for most of the genes. So it's just to make it easier to deal with these numbers as opposed to very, very small fractions. And the last thing I want to say is that, okay, you calculated FPKM, so now you have a normalized abundance, but it doesn't really, to me at least, it doesn't really make a lot of biological sense. So for example, it's not very intuitive. So let's say I got 17,000 FPKMs for this gene. What does that really mean? Okay, for every 1,000 bases, I expect to have 17,000 reads for this gene if I sequenced 1 million, if I mapped 1 million reads, but it's hard to digest, at least to me. It doesn't really mean anything. Okay, it serves well for me to compare control and experiment, but it's not really full of biological meaning. So there's been some improvement when people came up with this different way of normalizing, which is TPM. So TPM means transcripts per million. And in this case, it's much more intuitive in the sense that if I have 27,000 TPMs for the FGF21 gene in the control sample, what this means is that if I took from this sample, from this biological sample, a pool of 1 million transcripts, then I expect 27,000 of them to have come to be transcripts of FGF21. And to me, that makes a lot more sense. And you can actually think about this in a clearer biological way. So even better, when you normalize this way, you can actually have the same sum of TPMs for each sample. What that means is, here I'm showing you only for one gene, but if I took the entire table and I summed up all of the TPMs for the control sample, what I would end up getting is 1 million. And if I summed up the TPMs for the experiment sample, what I would get is again 1 million. So I put the two samples in the same standing. So they both have out of 1 million transcripts, how much would I see of each gene? So that makes it much easier to do direct comparisons across samples. And In the case of normalizations that account for both the transcript length and library sequencing depth, uh, like we discussed before, we can also compare across transcripts. So having 27,000 TPMs of FGF1 and maybe 50 TPMs of another gene means that I expect to find a lot more molecules of of FGF21 than of the other gene. And you can actually make those comparisons across genes as well. And it's great for identifying, for example, the highest expressed genes in your sample or the lowest expressed genes in your sample. You should not really be ordering that based on the counts because it doesn't take care of the transcript length, but you can do that with TPMs or FPKMs, where if you order all of the sequence genes or all of the quantified genes in your sample, you can actually identify the most highly expressed and the lowest expressed genes.
So I had a question.
Go ahead.
So there is this line in the R script that we have for the DE results where you set up the non-express cutoff at 10. So what exactly that 10 refers to?
Right. So I think Alper will go through it a little more when he talks about the different ways to set this threshold on using DE browser. But in that particular case, what you were selecting for 10 was on the counts. So
that's the raw counts?
Yeah, raw counts before normalization, and you were setting a threshold of having at least 10 raw counts. I personally do prefer to use normalized values to establish that cutoff. So after doing you have that option on the browser. Alper is going to show you guys. So after you do the normalization, then because you don't know if 10 counts is a lot for one gene, if the gene is short, and not for another gene, right? So that doesn't really get considered if you just do a cutoff on the counts. So I prefer to do set the threshold on the normalized counts, because that way you know for sure that that gene has a low abundance, and it's expected to have a low abundance considering its length and the depth you sequenced. So and even it makes the plot that Alper showed you guys of how many genes are not expressed mixed with how many genes are expressed. The histogram makes it easier to see the separation of non-expressed to expressed genes.
Okay, thank you.
No problem. So the TPM formula is different from the RPKM formula we were telling before, but it's still pretty simple. It is only taking each gene, for example, gene A, dividing the counts of that gene by the length of that gene. So no multiplying this time, it's just counts per base. And then dividing for each gene that fraction to the sum of all the gene fractions. So this is for each gene in your sample, you calculated this fraction as well, the ratio of counts per length, and you sum them all up. And then you divide the fraction of gene A by the sum of all the other fractions. This number would again be a very, very small ratio. And that's why we multiply it by 1 million to make more sense of it and to work with bigger numbers. So it would be hard to interpret. It would only be a fraction if we didn't multiply it. But now we can say out of every 1 million molecules, how many do you expect to see for gene A?
So just a quick question about counts.
Yep.
So is counts the number of reads associated with a particular gene, or is it is it the number of times the gene was identified in like the small reads?
So in the case of everything we've been saying here, that's kind of the same thing. So it is the number of reads you saw for that gene. We act like the number of reads we saw is corresponding to the number of individual transcript molecules, especially if you're working in the case of barcoded transcripts. So if you have a unique molecular identifier, then you can really be confident that one read came from one transcript molecule. But in this sense, we're just treating all the counts like they came from individual transcripts. So it is how many counts you assign to that gene, how many counts mapped to the locus of that gene in the genome, and you counted it as seeing that transcript one time in your sample. Does that make sense?
I see. So just as a quick like example of the FGF21 you show here,
Mm-hmm.
so you're saying that you found in the control group, you found 173 transcripts of FGF21.
Well, reads. We're translating it to transcripts, but it's not actually right unless you have unique molecular identifiers. So I'm saying it as in I saw 173 transcripts, but we know that each transcript is going to generate more than one fragment, and you're going to sequence those fragments independently. So you're actually going to have
this is
multiple.
it's more reads per million, not transcripts per million.
Right. It would be reads in this case because we don't actually have a correspondence of 1-to-1. Remember, we were talking about this in the quantification session. So if you assume a 1-to-1 correspondence, then you're saying transcripts. And if you have unique molecular identifiers, you can actually have that as correct association of 1-per-1. But in theory, in reality, when we're just working with regular RNA-seq library with no unique molecular identifiers, then correctly it would be more like reads in a million reads, but I don't want to say reads per million because that actually is another method of normalization, and reads per million only means that you normalize by the library depth. Okay, so transcripts per million. Yeah, transcripts per million measures the relative abundance and takes care of the length of the transcript. Counts per million or reads per million is just normalizing by library depth. So it's a different metric.
So when Neil was asking before about normalizing to the length of the transcript, the length of the transcript here is meant to do that. Is the idea that a longer transcript will be overrepresented?
Yes, a longer transcript will give you more reads, as well as a more abundant transcript. So if you want to say that FGF21 is more expressed than FGF1, then you have to be able to account for their length. So in this case, after normalizing by TPM or by FPKM, you can do that because length is in both formulas. All right, so that's it. If anyone has any more questions, feel free to ask. So when to use these normalized counts? That's also something that came up with when we were talking to Alper before, and he was showing you guys the process of doing the differential expression analysis, and he was working a lot with the raw counts. But in actuality, when do we use normalized and when do you use raw? The answer is pretty much all the time you're going to be using normalized counts. So for all sample and gene comparisons, so if you want to compare the same different genes in the same sample, what are the highest expressed genes in that sample? You need to use normalized counts. If you want to compare across samples, so do I have any genes with high fold change between experiment and control? Then you need normalized counts as well. So yeah, defining what are the most highly expressed genes, performing correlation analysis between samples. Ideally, that also has to be normalized because you need to account for the fact that you didn't actually sequence them at the same depth. And for doing clustering analysis, so I want to identify what are the most variable genes and then take those genes and actually perform correlation, clustering of my samples or clustering of these genes. You need to be able to account for the different library depth and the different length of the genes. For making the heat maps that you guys will see before, if you do it with unnormalized counts, you will actually see only a red blob and it's not really going to be informative. But when you normalize, you correct for the library depth and for the transcript length, and you can actually see differences across genes. So what about the raw counts, or the expected counts in the case of RSAM, which does a little bit of work on the raw counts? So that will only be used for the differential analysis input. And the reason for that is what I told you guys before. The count-based methods rely on the properties of the distribution of read counts. And that's why you can't normalize it before you give it to them. And you want to be able to effectively evaluate which genes show a significant change using these count-based models. So what are the normalizations that the differential expression analysis does? So even though you fed it raw counts, like I showed you before in the pipeline, and Alper talked about it a little bit too, you do, they do perform a certain normalization because they still need to account for the fact that you sequence samples at different depths. And that's basically all they will account for without the gene length. So they want to be able to compare the same gene across samples. And for that, you need to account for the library size. And one thing that is important to keep in mind is that for both of these methods, the count-based methods we've been talking about, DSeq and EdgeR, they make one big assumption about your data. And that assumption is that the majority of genes is not differentially expressed. So you may have outliers, you may have genes that have huge expression differences, and that's also something they want to account for. But most of them, more than 50% of your genes, will not change across your conditions. That's the big assumption they make because they rely on the depths of sequencing to be able to normalize each gene. And if everything is changing in your sample, then there's not really a good way to keep track of that. So, and yes, there is a way to get through this. If you, for example, have an experiment that you think you're gonna break this assumption and have the majority of genes changing, and that is using spike-ins again. So with spike-ins, you know exactly how much input you have in both samples, and you can do a normalization with respect to the spike-ins. But that's really a special case. And even in dramatic transcriptional changes, you're not going to see more than half of the genes changing. All right, so the method that DSeq uses for normalizing is called a median ratio. And what it means is just that you calculate the ratio of each gene to the depth of the library, compare it to a pseudo reference. So let me explain it clear with the little drawing. So basically what you do is you take the value of read counts across all your samples and you calculate an average and you create this pseudo reference. So it's a non-existent sample that is just a geometric mean of the read counts on all your other samples, okay? So this pseudo reference samples, then you calculate a ratio for each gene of the counts you saw in sample 1 to the counts you saw in the pseudo reference, okay? So then after calculating this ratio, you're gonna take a median for all genes of these ratios. And that median will be your library size factor. So basically this is the factor that you have to divide your counts by in each sample to make them all comparable. Okay, so it's per sample for each of these samples here. You're going to compare the gene counts to a pseudo reference sample, and you're going to then, for only the blue sample, calculate a median. That median is the size factor of the blue library. And you do that for each of your other libraries. So controls and treatments all treated separately, but they were used together to create the pseudo reference.
So can I ask a question?
Yes.
So it means you can compare genes between samples, but you can still not compare genes within samples.
Exactly.
Yeah.
Perfect observation. So with this normalization from DSeq, all it's doing is comparing the same gene across samples. It's not really doing any gene-to-gene comparisons within a sample. So that's why I can get away with this and not really normalize by the length, because if it did normalize by the length, it would lose the property of the counts that it needs to do the model.
Okay, thank you.
Elisa, so we need to summarize because
I think I'm almost done.
Okay, good.
I think I'm almost done, but I'll go a little quick. All right, so with TMM is another method called trim mean of M values. And that's the one that EdgeR uses for normalization. Again, it's only accounting for the library depth. So like Anne-Laure noticed, you can't really compare well across genes if you're using TMM normalization. You really should be comparing the same gene across samples. And what is an N value? So it's another ratio. It's just the counts of gene A in the sample X divided by the total counts in sample X. So remember that relative abundance, really simple, that we talked about in the beginning. But they divide that by the same ratio in a particular sample. So they pick, in this case, you pick one of your samples to be the reference, and all the others will be compared to that one. So instead of creating this pseudo-reference, it just picks one and compares all the others to that and generates these M values. And because you can have outliers, so you can have genes that will highly impact this because there's a big change of that gene across samples, your experiment and treated controls. You want to be able to remove those values. The way that EDGE-seq does it is by taking a median, so outliers won't have a big impact. The way that EDGE-R does it is by doing a trimmed mean. So it will get rid of these M values if they are on the top 15% or lower 15%, and it will only take a mean of the ones that are left. So it'll take care of outliers that way. All of this is to avoid cases where you have a dramatic change in a few genes from your experiment to your treated control or something like that. For example, in the LPS stimulation of dendritic cells, we see genes that go from having 0 TPMs to having 3,000 TPMs. So that gene is now becoming a significant fraction of your total reads. And if you just use the total reads as a method of normalization, you could be suffering from the impact of that one gene that really changed in its abundance. By doing these medians in this way of the TMM even better, you get rid of these outliers and you really are able to account for the changes across your library depth without suffering from their impact. And the last thing I want to briefly say is that you can also still have batch effects in your data. So normalization will not take care of batch effects. Even after normalizing for the library depth and for the gene length, you can still see batch effects that will bring your samples apart, and they are mostly due to different culture conditions, different sample and library preparation methods, and even having different individuals. So we can try to work around them if your experiment is well designed and the batch is not confounded with your test conditions. So I'm showing you here an example of our dendritic cell data. And in this case, we have maybe 4 donors here. So you have this D09, D01, and we have the G16 and G15. And these were sequenced in different times, but for all of them, we have the time point stimulation of dendritic cells. What we want to see is the difference in transcription before and after stimulation. But what it's showing, as you can see in this PCA, is that things are mostly clustering by donor. So you have all the D donors over here, and you have all the G donors over here because they were prepared largely in different times, and different individuals show this variation to begin with. So how do we handle this? So because it's not confounded with my experiment, so I have all the time points for each donor, and it's mostly the donor effect that I want to get rid of. So I can include the batch information in the DE models. This is something that I highly advise you to do, and Alper can talk about it later when you're explaining the browser. And you can also use algorithms to correct it. So last thing I'm going to show you is after using, for example, Combat, we fixed this entire effect. So now we can see that the samples are clustering largely by the time of the stimulation and not anymore by the donor from which they were generated. So if the effect is not confounded, you can get rid of it and you can overcome it. And really quick, I just want to leave this as a note. I don't even have to say it, but you guys should also go to IGV and set this normalization preference. So what this will do is that every time you load a TDF file, so a coverage file in your IGV, it will normalize your data by the library depth. So when you want to, for example, load multiple samples and see if you have different coverages, so you have higher expression of one gene, you'll see higher coverage for that gene. You should still account for the library depth, and this will do that for you. And it's only a one-time setting, and you never have to do it again. So do it now, and remember to do it if you download IGV in the future. And that's it. I'll leave you with the references, but you can check it on the slides later so we can switch Zoom meetings.